My name is Amanda, and today I'm going to be talking to you about an extremely important process known as DNA replication. So, looking at this 3D graphic here, you can see an original DNA molecule. And this DNA molecule is basically being split into two by an enzyme known as helicase. What helicase does, it basically unravels this original structure of DNA by breaking the hydrogen bonds between the bases of DNA. When this molecule is unraveled and separated into two strands, this strand up here is called the leading strand, and this strand down here is called the lagging strand. Where the original DNA molecule splits into two is called the replication fork. So the thing that we are witnessing right now is called the replication fork. This leading DNA structure, so basically DNA is anti-parallel, meaning that it runs in opposite directions. This leading strand up here runs from 3' to 5', and as a result, when it's replicating, basically this daughter strand here, it replicates from 5' to 3'. And since DNA naturally runs from 5' to 3', the replication in the leading strand is extremely simple. On the other hand, the lagging strand, the lagging strand is from 5' to 3'. As a result, when it's replicating, it replicates from 3' to 5', and that's when it gets a little more complicated, and I will explain that later. So, as I said before, let's take a look at how the leading strand replicates. So, as I stated before, it replicates in a 5' to 3' direction. And so here you have an enzyme known as DNA polymerase 3, which runs from 5' to 3', and it's basically synthesizing and assembling new nucleotides to the exposed DNA, which this exposed DNA basically acts as a template. It's the parent strand for the new daughter strand that is being synthesized. So it's assembling new DNA, and it's continuously assembling the new strand on top of the old strand. And so when it's assembling the new nucleotide sequences, it follows the base pair order, and this parent strand basically acts as the template. The way it acts as a template is adenine always binds with thymine, guanine always binds with cytosine, and as a result, the daughter strand on the top is able to read the template strand and basically synthesize accordingly. The lagging strand, however, is a little more complicated. So since DNA polymerase 3 only synthesizes from 5' to 3', and this lagging strand goes in the other direction, which is 3' to 5', it has to synthesize in a series of steps. So the first thing that happens is RNA primase, which is this enzyme, this pink enzyme here, basically synthesizes a primer on top of the parent strand. And this primer basically acts as a starting point for DNA polymerase 3. This DNA polymerase 3, which only synthesizes in a 5' to 3' direction, basically starts to synthesize DNA from this primer, which acts as a starting point, to the next primer. So it's synthesizing the DNA on top of the parent strand to the next primer. So this is DNA polymerase 3. After it has synthesized DNA from this primer to this primer, this entire section here of the DNA is called the Okazaki fragment. So after the DNA, these Okazaki fragments have been synthesized by the DNA polymerase 3, the RNA has to be replaced with DNA molecules, and DNA polymerase 1 does that. So it replaces the RNA molecule nucleotides with DNA nucleotides instead. So it replaces it. So this here is a DNA polymerase 1, and it's basically replacing the RNA molecules from the primer. And again follows the same base pairing, and it again is assembling on top of the parent strand, which serves as the template for the new daughter strand. So after that, you can see this little gap here. It has to be filled, and so DNA ligase basically connects these two primers, these empty sections together. And as a result, when you go back here to see the DNA molecule as a whole, you can see that this entire process is semi-conservative, because when it is all done, which this video doesn't really show, but when it is all done, you have the parent strand remaining with the daughter strand on top, and so the original DNA, half of it is there, and as a result, it is known as semi-conservative because the parent strand serves as a template for the daughter strand to replicate. And in your cells, in your body, DNA has multiple replication forks, and this makes it extremely efficient. And that's why DNA is being synthesized in your body all the time. And that's DNA replication. Thank you for watching.